Hi, everybody. My name is Chris. I work for Grand Canyon Conservancy Field Institute. I'm here with Ann Miller, who works at Science Resource Management at Grand Canyon National Park, and she's got some really cool things to share with us. So without further ado, Ann, take it away.
Okay. So first, I'm just going to talk a little bit about myself. I'm the geologist / paleontologist at Grand Canyon National Park. And I actually started as a paleontology assistant at Glen Canyon National Recreation Area in 2015, then moved to Grand Canyon National Park as a paleo intern in 2016. Now, since then, I received my Master of Science in Geology at NAU while continuing to work for the park. And in fact, my thesis had to do with the Bright Angel Shale Formation, which is one of the formations in Grand Canyon. So finally, just last year, I became an official government employee. And these are just a few pictures of some of my adventures at these two parks. Our Geosciences program includes hydrology, karst, caves, geohazards, air quality, and my personal favorite, paleontology. Now, the main purpose of the hydrology program is to better understand Grand Canyon's complicated aquifer system. This is just a diagram that generally shows how water moves through Grand Canyon rock layers, beginning with the source sinkholes on the Kaibab Plateau, where water sits into the sinkholes and infiltrates the ground from there. Now, there are two main aquifers. There's the sea aquifer, where water comes out at the Coconino and Hermit contact. And then there's the R aquifer, where water comes out of the Muav limestone. Now, as you can see, a lot of times these are often connected aquifers. And the entire system creates this labyrinth of pathways and caves, especially in the Red Wall Formation. Now, one thing to note is that all streams that lead to the Colorado River in the Grand Canyon are sourced from this aquifer system. So all streams begin with a spring. Now, in Grand Canyon, there are over 600 springs. There are several important reasons for characterizing this aquifer system. Climate change is one of them. Climate change poses a risk to reducing the water source, but not just reducing our drinking water, it also poses risks to the stream ecology. For example, native fish, such as the humpback chub, rely on these springs to maintain their habitat. So if the water source is reduced, then you have reduced flow and you have reduced sediment transport. And overall, you're affecting the native fish habitat and you're reducing that habitat. So that is very concerning to us as well. So there are plenty of reasons for us to look into this aquifer system and understand it in order to protect these really important resources. There's also potential vulnerabilities to the source contamination. So we're talking on the Kaibab Plateau, bison, fuel truck accidents, wastewater treatment plants. Those are all concerning. Visitation, of course, at Grand Canyon is always increasing. And then with the added pipeline reconstruction, this aquifer system is in need of more attention than ever. So right now, both the north and south rims get their water supply from Roaring Springs, which eventually feeds into Bright Angel Creek. And this is a picture of Roaring Springs coming out of the canyon on the right, and on the left shows in the Roaring Springs cave, our hydrologists are measuring discharge. Now, the new Trans-Canyon Waterline project plans to pull water from Bright Angel Creek at Phantom Ranch, which will also take water from several different springs that feed into the Bright Angel Creek system. And so our regular spring monitoring in this watershed will just be ramping up a little bit. So this includes installing new gauges at Bright Angel Creek springs, such as Wall Creek, Angel Spring, Transept Canyon, and Phantom Creek. we have gauges at some of these other springs already. So these are ones that will be new gauges that we're adding to our data. Climate change studies will also continue in order to predict impacts to Grand Canyon's water supply, and eventually how it can affect the stream ecology for native fish. And continuous data collection and monitoring at these springs will continue before, during, and post pipeline reconstruction to see what changes have occurred. And Grand Canyon will continue to manage 4 additional gauge sites in our aquifer springs. So remember, that's the one coming out of the Moab limestone. And these include Basie's Paradise, Mystic Falls, Modred Abyss, and Tapete Spring. So we collect the same data there for the same purposes at those sites, but we only visit those ones on an annual basis due to access difficulty, which makes sense. It's the Grand Canyon. So moving up to the source of these springs is the extensive karst system on the Kaibab Plateau. Now, in this case, karst refers to the presence of sinkholes, similar to what this person fell into. And sinkholes are depressions or holes in the ground caused by some sort of collapse due to the dissolving limestone beneath the surface. So you're getting this hole dissolving, and then somehow something collapses and allows the hole to open up. And so instead of water sort of flowing along the surface, water ends up flowing into these sinkholes and infiltrating the ground. And then it travels through these pathways in the aquifer system, eventually coming out at spots like Roaring Springs. Now, according to our data collection, there are about 6,973 identified sinkholes on the Kaibab Plateau. Now, sinkhole mapping provides us with a karst vulnerability map. So shown here is a map of sinkhole density on the Kaibab Plateau. You can see that the red marks the higher density of sinkholes, and it happens to be along the main highway entering into the park. So these sinkholes are known to be vulnerable to potentially contaminating drinking water based on a dye trace study that occurred from 2015 to 2017. So during that study, various environmentally safe dyes were injected into several large sinkholes noted by these circles. So a different type of dye was put into these sinkholes in order to track where that water came out of. And what we found was that the dye followed known fault systems. So these black lines here are major fault systems. There's a lot more to it, but these are just some of the major ones that have been mapped. And the dye came right out at Roaring Springs and in other springs in the Bright Angel Creek system. In addition, some of the dyes also went towards the east, followed certain pathways there towards Basie's Paradise and came out there. In addition, it also came out towards the west in a different pathway towards Tapetes and Deer Springs. So it's a very dynamic system. And we plan to continue this dye trace project in the coming years, but it's funding dependent. So we couldn't do it this year, unfortunately. Now, caves are created by this water flowing through pathways and dissolving limestone. And there are over 300 identified caves in Grand Canyon, and likely more to be discovered. Now, all caves in the park, with the exception of Cave of the Domes, were closed to the public in the 1980s due to increasing damage of fragile cave resources. Now, I should make a note here that Cave of the Domes is currently closed right now, and I'll go into that a little bit later. Of course, many people still try to access these caves, and there's been signs of some damage still occurring. But overall, we're asking people to, if they're entering caves, they have to be a researcher, and they have to have a research permit in order to enter a cave. And so it's really imperative to protect these caves because they contain many different significant resources. Now, certain caves contain world-class cave formations, such as these extremely rare and fragile gypsum formations. Now, these are actually endemic to Grand Canyon, which means you're not going to find these formations anywhere else. And in fact, scientists are still trying to identify them. So these are extremely important to us. If we let any caver go in there, it's likely that these will just fall apart. So a lot of these caves are extremely fragile and very significant. Here's just some other pictures of neat formations that are also very fragile. In addition to these fragile cave formations, there are also fragile and significantly well-preserved Pleistocene age fossils, such as this extinct American cheetah, the extinct Shasta ground sloth, and the extinct Harrington's mountain goat. Only to name a few. There are many, many more that have been discovered in Grand Canyon caves. There are also mummified bats and ringtail cats. And of course, there are significant cultural resources like split twig figurines, baskets, pottery, and others. The stable cave environment allows these resources to maintain their preservation. Now, going back to Cave of the Domes, this was the only one that has been open to the public, but since June 2020, there have been concerns about white nose syndrome and, of course, COVID-19 spreading from cavers to bats. So this is a temporary closure. Now, to talk a little bit about white nose syndrome, it came about in 2006, and it has devastated bat populations across the eastern US, and it's rapidly spreading towards the west. White nose syndrome is caused by a fungus that's native to Eurasia that can be passed from bat to bat, or in some cases from dirty caving gear. So it can even jump states or continents because of that. But the reason why it's deadly to bats is because it actually causes irritation. And so it wakes up a bat that's hibernating and forces them to use energy reserves that they don't have enough of. And so they use this energy too rapidly and end up dying because of that. Now, we don't yet have documented cases of white nose syndrome in Arizona, but likely in the next few years, it will come. Sarah Haas will be talking a little bit more about our status with white nose syndrome. Now, right now, cave researchers with a permit are currently asked to wear N95 masks in caves and to keep their distance from bats. Now, there's exciting research being conducted in Grand Canyon caves. For example, NAU is working on genetics of mummified bats because there are a lot of them and they're very, very cool. Also, a PhD student from NAU is working on a 3D model of the Kaibab Plateau hydrologic system using LiDAR. This will take a while, but this is a very interesting project that will really help us understand the different pathways that this water is traveling through the rocks of Grand Canyon. So we're really excited about that. In addition, scientists at the Kentucky Geological Survey are working on a history of cave formations in Grand Canyon. And then, of course, our park staff work with cave researchers who have a permit to continuously find and survey new caves. All right, so we do have a geohazards program, but it's been unstaffed for almost a decade. And I'm finally here to revamp the program, and we're starting to make some progress. So in 2013, interns documented past and present mass movement events, like this rockfall here from February of 1993. This is in Phantom Ranch, and this rock hit one of the bathrooms. And they mainly focused on the corridor area, so Bright Angel Trail to North Kaibab Trail. And I've been assessing this data, working with the Geologic Resources Division in the regional office to implement the Unstable Slopes Management Plan, which is just a comprehensive method for mapping geohazards and estimating risk. And so we also plan to do a geohazard assessment in the Phantom Ranch and Supai Gardens area, where people and facilities are most vulnerable to these rockfalls. In addition, we will be receiving new LiDAR data that we're all really excited about, and we're hoping to use this data to document and potentially model geohazards in the future. All right, we also monitor air quality on a weekly basis, as mandated by the Clean Air Act of 1970, which gives a special air quality and visibility protection to national parks larger than 6,000 acres. So that makes Grand Canyon National Park a Class 1 airshed designation, which is great because that allows us to ultimately improve visibility and protect our amazing views. So we have 2 sites along the South Rim, and we continuously monitor the amount and chemistry of precipitation, the concentration and chemistry of particulates, and of course, ozone and visibility. Now, air quality at the park can vary on changes in weather, smoke from fires, and of course, anthropogenic pollution. So it's really important that we regulate this air pollution. And we've been doing it since the Clean Air Act in 1970. And what we have found is that the air quality is actually starting to get better. So here's a diagram that shows sulfate concentration levels decreasing into 2020. So that's good news. In addition to that, we're also showing increased visibility going into 2020 as well. So because of this monitoring, things are actually improving as far as air quality goes. All right, I will finish off this presentation by talking about our paleontology program. Now, back in 2019, I did a GTS presentation on the various fossils that we find throughout Grand Canyon rock formations. But this time I will discuss more about what we're working on and then go over a few spots along trails that you can actually show fossils to your clients. So I've highlighted a few things that we're currently working on. Of course, it's not everything, but it's some of the exciting things I'd like to talk about. And this includes ongoing surveys surrounding the pipeline, Cambrian research, photogrammetry, and Permian research. So we've been doing in-depth paleontology resource surveys along the waterline corridor in order to document significant paleontology sites that may be vulnerable to damage during construction of the project. So we will be present during certain areas of construction to make sure that some of these fossils aren't damaged. So getting them inventoried is really important to know where the significant ones are and how we can protect them. So here's some examples of some fossils that we find near the waterline. Here's a picture in the middle of me pointing at the water line. You can kind of see it. It's just in the bushes a little bit. And to the right is the same area. The pipeline's still right here, but I'm over here pointing at an entire rock outcrop that is just full of these burrows, which are in situ, meaning that they're within the bedrock, which is important for the context for scientists and geologists. And we're finding tons of these burrows among all kinds of other traces here. So this is a very important spot that we don't want to be damaged. We also see an example of some of these loose rocks surrounding the pipeline. And if you look very closely, you can actually see that they're just chock full with these marine invertebrate fossils, including this really nice crinoid ossicle impression. All right, so there's a lot of Cambrian research going on in the park right now. So I can only talk about a little bit. We're working with scientists from various universities and other affiliations, all led by Carl Karlstrom, who is likely watching this presentation. This work involves dating the Tonto group using detrital zircons, chemostratigraphy, and of course, trilobites. And throughout this work, we have actually found many new trilobites with expert Fred Sundberg, shown here looking for trilobites. And this includes a potentially new species of Albertella that we found just last year. So there's one species of Albertella that is known to the park, but this one is appearing to be a little bit different. So it might be actually a new species that Fred is trying to identify. So we're very excited about that. In addition, I'm working with paleontologist Dave Elliott from NAU. He was also my thesis advisor. And we're working on a paper to describe these Cambrian arthropod tracks that we found, again, just last year. The morphology of these tracks suggests that it was made by a large benthic arthropod called Tigopelti gigas. And because this arthropod lacks segments, it actually has some trouble turning its body as it walks along the surface. So it ends up making these 4 to 5 point turns where it has to stop, kind of like when you're trying to parallel park. And so this is actually a very unique trait to Cambrian arthropods. And so although Tigopelti specimens haven't been found in Grand Canyon specifically, in fact, they've only been found in the Burgess Shale, which is of similar age. These tracks suggest that it was likely present during the deposition of the Bright Angel Shale in Grand Canyon. So these tracks kind of give proof that this arthropod was likely around at that time. So very cool stuff. Another cool thing that we're working on is processing digital elevation models through photogrammetry for vertebrate trackways in the Coconino Sandstone, many of which are significant in situ localities, like this model here on the bottom. In situ, again, meaning it's within the bedrock. So it happened where it happened, which is very important for context. And these models really help to reveal details that you can't really see from pictures alone or even in the field. For example, this track block up here shows what we think is the claw of this reptile. And this was actually very difficult to see when we were in the field and also difficult to see in photos. But having this digital elevation model really makes them pop out and we can see these features very nicely. So we'll be working with several different scientists on the Coconino tracks, as well as the invertebrate tracks too, to help identify them and hopefully conduct a reconstruction of the paleoecology and paleoenvironment of the Coconino Sandstone. All right, lastly, before I talk about fossils you can find, I want to talk really quick about our Permian research going on. Now, since our recent discoveries of these well-preserved plant fossils in the Hermit Formation, we've started to work with UC Berkeley paleobotanists on identifying and reconstructing these fossil plants. Now, keep in mind that the last major study on the Hermit Formation, especially regarding to plant fossils, was conducted by David White in 1929. So we're well due for a study on the Hermit Formation. And since then, we've been finding so many new plant fossils that have yet to be identified. These two on the top, we're still not quite sure what they are. So hopefully a new species comes out of this as well. But what we've really discovered here is that Grand Canyon is quite unique when it comes to hermit plant fossils. It's not the best preservation, as you can see, but what we're seeing is actually somewhat of a three-dimensional picture of what these plants might have looked like. And this is something that a lot of other localities lack. Oftentimes, leaves will come off the plant, come off of the stem and fall to the ground, and that's how they're preserved. So you get a preservation of a leaf, a preservation of a stem, and different parts of the plant. And paleobotanists tend to have to try to put that together. But what we're seeing in Grand Canyon is that this is already put together. So in this picture here on the bottom, you can see a stem, an impression of a stem here. And there are actually fronds that come off of that stem. And even up here towards the left, there's another frond impression that's oblique to the bedding plane. So you can't see it very well, but it is also coming off of the stem. And this is just one example of what we find. We're finding a lot of these in situ fronds connected to other fronds and other stems. And we're able to see sort of the full picture of what these plants look like. And so this is very exciting for the paleobotanists at Berkeley because they've never seen this before. And so this is how Grand Canyon paleobotany is actually quite unique. And so we're very excited to see where this research takes us. I should also mention that the reason why this is preserved that way is because these plants had to have been buried in place while they were still put together. So there had to have been some sort of rapid sediment burial in order to get as much of the full plant as you can preserved. And that's not always the case, but when you find it, it's very exciting. Okay, so now I'm just going to discuss 3 localities that you can take your clients to show fossils. This isn't something we normally do, so we decided that these were the localities that you can share with other people. We really want people to appreciate these fossils and to understand their significance and why it's important not to vandalize them. So we're hoping that showing you these will sort of help your visitors to understand the importance of these resources. So the first locality is along Hermit Trail, and it's just along the first few switchbacks near the Hermit Trailhead. And if you follow the sort of benches of the Kaibab limestone, sort of off the trail, and you look closely at the rock, then you will see all kinds of marine invertebrate fossils like sponges, bryozoa, rugose corals, and crinoids. And so there's some things you can talk to your clients about them. For example, sponges. People don't tend to get that excited about sponges, but you'll know it's a sponge when you see sort of a concretion with this sort of brainy like texture inside of it. And that's the internal structure of the sponge. But what's really cool about sponges is that they're among the simplest and earliest multicellular organisms to evolve on Earth. And they've still managed to remain successful today. And so they attach themselves to the ocean floor, and they actually inhale their food from the outside of their bodies, as you can see by this dye. So they inhale their food and then exhale their waste out of the opening called the osculum. So just kind of a cool trait about sponges. And this is essentially why sponges like to have a stronger current, because they can get more materials, they can get more food with more things coming their way. They don't like murky water because it tends to clog them up. Now, bryozoans are pretty cool to me. You'll see a lot of these in that area, in the Kaibab limestone. Now, a bryozoan fossil is somewhat of like an old abandoned apartment complex in which millions of individuals once thrived together. So each little apartment or zoachium housed a tiny little organism. So these are the little apartments, these little holes or windows, and they housed an organism called a zooid. And each zooid pops out of its apartment to filter feed on plankton using its tentacles, and then it'll pop back in. So these are just really cool, and you'll see a lot of these in that area. And then lastly, just showing some rugose corals. These are a little bit harder to find, but when you find them, they're very exciting. They're also known as horn corals because they look like a horn. And they also attach themselves to the seafloor and use their tentacles to capture microscopic materials. Okay, the second locality is also on Hermit Trail in the Coconino switchbacks. So some of you probably know where these historic steps are, but perhaps not everyone has noticed the plethora of trackways all in this area. So just to show a few, you can see the stairs are up here. So looking up towards the stairs, you can see all kinds of trackways. There's this huge one here that isn't very well preserved, but it's large and you can see a tail drag. There's also this one here that decided to change directions. That's one of my favorites because I always wonder why it changed directions. And then these ones here on the right, you can see actual claw marks from some of the reptiles, and even some invertebrate tracks from spiders, prehistoric spiders. So here's a little bit of a better view. This is that big one that I was talking about with the tail drag. So you can see the footprints a little bit. It's not the best preserved, but it's really cool when you can see it in person and imagine this huge creature walking up a sand dune. In addition, these little tracks are actually on a brand new platform that Trail Crew just put in right at the top of those steps. And then there's these just a little bit off the trail. They're just really cute. And then this is a close-up of the spider tracks. And something to note is if one of your visitors asks, well, how do you know the difference between a spider track and a scorpion track? Well, the main difference is that the spider tracks tend to have these little impressions of clusters of 4, sometimes 3, it doesn't always get preserved, next to each other. So you have these little clusters of 4, but with the scorpion, you actually have clusters of 3 that are more at an angle. And so they're actually quite distinct from each other. These are just pictures of modern day spider and scorpion. Okay. And then lastly, a lot of you probably already know about this locality, but I wanted to share the vertebrate tracks in the Supai group along the Bright Angel Trail. So this is just a little bit below the 3-mile rest house. And these are, so the tracks are actually facing up trail. So it's a lot harder to notice the tracks when you're going up. It's easier to notice when you're hiking down. And these are actually based off of a paper that was recently published by Roland et al. In 2020. And so far, they're the oldest vertebrate trackway reported from Grand Canyon from 313 million years ago. So far, meaning we've already started to find more trackways that may be a little bit older or of the same age. We just haven't quite gotten the research yet and reported it yet. But this is the first one, so it's very exciting and very accessible. Hope you can stop there and show your visitors that these were made by a reptile, and likely one of the first reptiles to start evolving. All right, so that's pretty much it. I just wanted to end with sharing our social media presence. We have a Facebook called Grand Canyon Paleontology and an Instagram, and it's not NPS related, so it's sort of a fun thing that we post our research. We'll post cool fossils we find and some funny memes and different things like that. But it's really a great way to keep up with what we're doing in the paleontology program. So with that said, any questions? And I just had to put this slide because I know a lot of you have visitors that don't know where they are or what they're doing. So I just thought this was kind of funny.
That is funny. Thanks, Ann. That was really great. Really appreciate all the wonderful insight. I certainly have lots of questions that I can't wait to ask you. For all of those who are watching, don't forget to tune in the 26th of February, 9:30 to noon, where Ann will be there along with all the rest of our speakers to answer all your questions. Don't forget to, if you can, submit your questions ahead of time so we can line it all up and make it as efficient as possible. With that being said, see everybody on the 26th, and thanks, Ann. Really appreciate all the great knowledge and insight.
Thank you so much.